So hello, everyone. Thank you very much for joining us today for the BC Speaker Series in January. This is our international webinar series that has been organized by BC. So just a little bit about BC. We are the Center for Bee Ecology, Evolution, and Conservation at York University, and we are an organized research unit. If you'd like to learn more about us, you can visit our website, yorku.ca/bees, or feel free to email me, the BC coordinator, at bc@yorku.ca. We are an initiative that strives to advance research in the field of bee ecology, evolution, and conservation. And very briefly, the mission of BC is to foster interdisciplinary, innovative, collaborative, and cutting-edge research. This research is then used for the advancement of knowledge and the implementation of policy changes to help sustain pollinators globally. Our overall goal is to apply our collaborative efforts to the development of policies and environmental management for the long-term sustainability of bees and the vital ecosystem services that they provide. So if you'd like to hear more about BC, please feel free to join our listserv. So if you send an email to listserv@yorku.ca with the following command in the body of the email, subscribe bees, and then your first name and your last name, you'll be kept in the loop about all of our programming. You'll receive a monthly newsletter from us, as well as reminders about events such as our BC Speaker Series talks like the one today. And now, if you are a York University researcher or collaborator, please feel free to contact me about more information about potentially applying to be a BC associate. And you can see below here, we have a collection of our current BC associates, some nice photos of them out doing some research or pinning some insects. So for today, you are joining us, of course, for the BC Speaker Series. Generally, we run this program one Friday each month, typically the second or third Friday of each month at about 11 a.m. Eastern via Zoom. You can see that we've already had a wonderful selection of fall semester programming. Many of these talks are available for viewing on our website as well. And we have an additional 4 speakers lined up for our winter semester starting today. And then we have a speaker in February, March, and April. If you'd like to learn more about the series, please feel free to visit our website. And on this website, you can also register for all of the future winter sessions as well. Now, a few logistics for today. We are, of course, using a Zoom webinar platform. So only panelists and hosts can have their cameras and microphones on. You can use the Q&A box located at the bottom of your screen to pose questions to the speaker. And at the end of their presentation, we'll pose the questions back to the speaker and have them answer. Please use the chat box sparingly just for general comments, as it can be distracting for the speaker to have notifications continually popping up. And if you do have any trouble, feel free to send a private message to me or to email bc@yorku.ca. Now, this presentation will be recorded and posted. So if we do run a little bit over time with the Q&A session at the end and you have to leave at the hour mark, don't worry, both the presentation and the Q&A discussion are being recorded and will be posted. Now, speaking of our YouTube channel, we do have several previous talks posted from previous webinar series, like the BBST series that we ran previously, as well as the Associate Speaker Series and several talks from past BeeCons, including videos from our most recent BeeCon. We invite you to check them out. And of course, more videos will be posted over time. So we invite you to subscribe to our channel so that you will be notified when our next video is posted. Before we get started today, I would like to recognize that many Indigenous nations have long-standing relationships with the territories upon which York University campuses are located that precede the establishment of York University. York University acknowledges its presence on the traditional territory of many Indigenous nations. The area known as Takaranto has been caretaken by the Anishinaabe Nation, the Haudenosaunee Confederacy, and the Huron-Wendat and the Métis. It is now home to many Indigenous peoples, We acknowledge the current treaty holders, the Mississaugas of the Credit First Nation. This territory is subject of the Dish with One Spoon Wampum Belt Covenant, an agreement to peaceably share and care for the Great Lakes region. If you would like to know more about the history of land that you come from or where you are joining us from today, this website, native-land.ca, is a fantastic resource for exploration. It was initially a Canadian initiative, but now it has worldwide coverage. So you can look at such locations as where you complete your research, where you collect your samples, etc. And now I would like to invite Dr. Sandra Rehan, the director of BC, to introduce today's speaker.
Hello. Very happy to introduce Dr. Olivia, and very happy to learn that she became a new assistant professor this year. So she's just started her lab at the University of Wisconsin-Eau Claire. And my understanding is that's home for you, right? Wisconsin is where you originated. So she's come full circle, much like myself. We did a global tour to end up back where we started. But yeah, she did her postdoc most recently at University of Wisconsin in Madison. But before that, she went all the way over to Australia and did her PhD in Sydney at the Hawkesbury Institute, where she worked with some colleagues of mine. So I've known Olivia for a little while, and she did excellent work there on alidapine bees and foraging ecology, some agroecology, kind of a whole lot of cool stuff. And then before that, she did a master's in Maryland in entomology, and then before that, she was originating again from Wisconsin, Madison. So she's a Wisconsinite or whatever they call themselves. And so it's cool to see the culmination of a really long legacy of work. Her work transcends global change biology and pollination biology, looking at plant pollinator interactions, and some stuff even in sociobiology, looking at casteless societies of the alidapine bees. Today, probably talking more about the global change biology and pollination ecology work, but whatever she touches, it's very interesting, and it's been pretty exclusively bee related. So she was a perfect candidate for today, and we're very happy she had the time to share her work with us. So with that, thank you very much, Olivia, and take it away.
Great. Well, thank you very much for that lovely introduction. I am excited to talk to you all about some of my work. I'm actually not going to talk about any of the work I did in Australia or my work on alidapine bees, but I'm happy to answer questions about that later. That's some of my favorite research I've had the opportunity to do. So I'm going to go ahead and get started with my acknowledgements. I think, research really is, the product of a village. There are lots of folks that go into making research happen. And I like to start off with my thank yous because the work I'm about to tell you about wouldn't be possible with these folks. And then I'm not rushing at the end to sort of squeeze it in. So I'd like to start by thanking and acknowledging the traditional owners of the land where I live, work, and do my research, the Ho-Chunk, the Sauk, and the Kickapoo peoples. I'd also like to acknowledge my postdoctoral supervisor, James Krall, and all the other folks listed here that really helped to make these projects come to life. So I probably don't need to give a whole lot of background on pollination. This group probably is pretty familiar with pollination and that sort of plant-insect interaction, but just so that we're all on the same page, pollination is a mutualism between flower visiting insects, largely bees, and flowering plants. And so here's an example here where we have a honeybee visiting a cranberry flower to collect pollen and nectar resources that she'll take home and use to provision her sisters. The cranberry plant also will benefit as it's able to reproduce, right? So pollen gets moved from flower to flower and it's able to produce cranberries, allowing the plant to reproduce and allowing those of us that like to eat fruits and vegetables like cranberry to have a delicious bit of food to eat as well. So my work largely thinks about how aspects of global change, both abiotic and biotic factors, are influencing pollinator biology, pollination services, things like that. And so some of the abiotic factors that I've considered in my work include atmospheric CO2, so elevated atmospheric CO2, warming temperatures, as illustrated here with this map of Wisconsin over time, and land use change. And here I'm thinking land use change in going from, habitat. So maybe this was a forest and now is a city. It's habitat loss for many insects as that city, that man-made landscape really is no longer a valuable resource for many insects. And so that's sort of an aspect of global change, urbanization, and habitat change. But there are many other biotic factors too that can influence the way that pollinators live, including things like invasive species that can come in, like this garlic mustard, and change the composition of the community there. The spread or the introduction of new and more virulent pests and pathogens, like we can see with this bee with honey, or a honeybee with deformed wing virus, but then also habitat change from more of a biotic perspective. So here I'm thinking, they've bulldozed this land down to nothing, and perhaps they're getting ready to build a park. And so maybe it went from forest to parkland where it still might have some value to pollinators, but it might be degraded in quality. So there might be flowering plants, but maybe fewer nesting opportunities and whatnot. So today, I'm going to talk to you about sort of three of these, some of my work on elevated atmospheric CO2, temperature, and then habitat change and temperature together, sort of thinking about thermal microhabitats within the landscape and how that influences pollinator foraging behavior. So as I said, we're going to start with some work I've done on elevated atmospheric CO2 and how that affects pollen nutrition, thinking about temperature and foraging, and then shade microhabitats on foraging. So to get started in the sort of atmospheric CO2 realm, I like to show this graph, and I think probably most of us, again, have seen something like this, or maybe even this exact figure, but what this is showing is really that over time, atmospheric CO2 levels have been rising. And if we look back 50, 100, 150 years ago, they've just been pretty steadily increasing ever since. And this graph goes to 2018, but this would continue this trend up until 2025. And so atmospheric CO2 is rising. It's something that's maybe talked about a little bit in the news, but one of the ways that this change in atmospheric CO2 is important is that elevated CO2 in the atmosphere affects plant growth. So if we think back to sort of intro bio, when we're thinking about photosynthesis, carbon dioxide is a building block for plants, right? So we, on the other hand, are going to take in oxygen and we are going to breathe out carbon dioxide, and plants do the reverse. They take in that carbon dioxide, use that carbon as their structural component. That's where they get carbon to grow and to build leaves, and then they release oxygen. And so when you have more carbon dioxide in the atmosphere, basically what you're doing is fertilizing the plant. So if you think about houseplants or maybe your fruits and vegetables in the garden, you might need to provide them with fertilizer in the forms of nitrogen or potassium or something like that you would mix into the soil so that the plants have enough nutrients to grow. And so having this elevated atmospheric carbon dioxide basically acts as carbon fertilizer for these plants as well. So they have more carbon available to them. And basically what it does is it helps plants grow bigger. So we know that elevated CO2 can lead us to have taller plants. So this man here is standing next to some conifer trees at ambient plus 150, plus 300, and plus 450 parts per million CO2. And you can see that those trees get substantially taller as the amount of CO2 available to them goes up. And we know from other work that elevated CO2 can affect plant leaves. They can grow bigger, they can grow thicker. It can affect flowers. For example, flowering time, they may flower earlier or have smaller flowers. But what's less understood is how elevated CO2 might affect flowering resources. So, as somebody who works with pollinators, I think a lot about pollen, I think a lot about nectar. And so what we don't really know is how elevated CO2 might affect these floral resources. And so there's a study that came out in 2016 by one of our collaborators, Lou Ziska, that looked at Canada goldenrod and elevated CO2. And so his group did two things. First, they looked back in time when CO2 levels were lower. So they looked in the historical record using herbarium samples going back about 150 years to see if the protein available in Canada goldenrod pollen changes over time, which really is a change in CO2 in CO2 levels. And so they looked back in time to see, 150 years ago, was there more pollen available in Canada goldenrod protein? And then they also in modern time did an experiment to experimentally alter these CO2 levels to see, if we change the CO2 levels available to this plant, how does their pollen protein respond? And here's a figure of what they found. And so the pattern was the same across both experiments. So I'm just going to show you one. But along the horizontal axis, we have atmospheric carbon dioxide ranging from about 280 to 400 parts per million. And then on the vertical axis is estimated protein percent in the pollen. And so across both studies, they found very consistent results that as carbon dioxide in the atmosphere goes up, pollen protein in this Canada goldenrod goes down. And so this is sort of one of a kind study, right? There aren't very many other studies like this. We don't really have other plants to compare this to and to figure out whether this is sort of a fluke of Canada goldenrod or whether this is a trend across all flowering plant species. And this sort of got me thinking, if you are a flower visitor, like a bee or a butterfly that relies on pollen, that's your source of protein. That's where you get the building blocks for yourself. And you don't really have other places to go for this source of nutrition. And so, think if we went to the grocery store and our tofu or our steak or our chicken was suddenly half as full of protein, that would be a problem, right? We'd have to eat more, we'd have to buy more, we'd have to work harder to make sure we're maintaining that level of nutrition. And so for pollinators, it's likely the same, that if we're seeing this kind of widespread decline in pollen protein, then they're going to have to work harder for their nutrition, and they might have to switch their foraging preferences, which could lead to knock-on ecological effects. So plants that have perhaps less of a reduction in pollen protein might be favored over those that have a stronger reduction in pollen protein, and that could restructure plant pollinator interactions as well as the communities of both of these important organisms. And so I specifically was interested in what about other, flowering species? Is this sort of a widespread trend, or is this sort of a fluke for goldenrod? And so this is work that I did in collaboration with Anupreksha Jain, who started this work at Harvard and then continued it together with me at the University of Wisconsin. And so we evaluated pollen nutrition across 12 flowering plant species. And so we used percent nitrogen as a proxy for protein. So since protein is a really highly nitrogenous compound, we can basically estimate protein content from this percent nitrogen in the pollen. And so Anupreksha carried out the first experiment at Harvard with 6 flowering species using a 450 parts per million CO2 for the ambient CO2 level, which was ambient in the Boston metro area where Harvard is, and then elevated CO2 about 200 parts per million to 650 for the elevated CO2 treatment. I did a second follow-up experiment at UW-Madison using 7 flowering species. We both had sunflower in our study, so that's why it's 12 total. Ambient CO2 levels in Madison when I was conducting this study were about 400 parts per million, and we elevated our eCO2 treatment up to about 690 parts per million. And we measured a whole bunch of other things beyond pollen nutrition, growth, leaf number, flower size, whatnot. I'm just going to talk about pollen nutrition for today, but if you're interested in the rest of that, this paper came out about 6 months ago in Scientific Reports, and you can find the rest of that information there. So when we look at what happened with pollen percent nitrogen, I'm going to start with experiment 1. What we found in experiment 1 was that there were no effects of carbon dioxide treatment on pollen percent nitrogen. So here we have our 6 flowering plant species across the bottom, and then for each plant species, we have a violin plot on the left in gray, which is the ambient CO2 treatment, and a blue one on the right, which is the elevated CO2 treatment. And so for these 6 species, it does not appear that we're seeing any effects of elevated CO2 on pollen protein. When we look at experiment 2, the one that I did at the University of Wisconsin, we find pretty similar findings. Again, the only species that showed any significant response to CO2 treatment was buckwheat. And interestingly, it had an increase in pollen protein under elevated CO2. So I guess really the positive take home from this is that what was seen in goldenrod potentially is a fluke and is not something that's happening across the board, or it might be something that's happening in a subset of plants, but it does not appear that elevated CO2 is going to lead to sort of a broad decline in pollen protein. So that was a pretty exciting finding. And also really not what we expected. So we sort of assumed going into this study that plants would produce pollen with lower protein under elevated CO2 because nitrogen would become a limiting nutrient. And we know that in other plant tissues like leaves and stems that there is a reduction in nitrogen under elevated CO2 across many plant species. And so we were pretty surprised to find that there was no effect of elevated CO2 on pollen percent nitrogen. One difference between our study and the ZISCA study on Canada goldenrod was fertilizer levels. And so in the ZISCA study, they looked at plants that were grown in the wild and that were grown without any fertilizer. But in both of the studies in Harvard and at Wisconsin, we used a little bit of light fertilizer when we initially planted because we were growing plants in a greenhouse in sort of a limited volume capacity. And so we needed to make sure they had a little bit of fertilizer to be able to continue growing in a pot. And so we were interested in sort of trying to understand whether or not the role of this nitrogen fertilizer was perhaps obscuring any findings we might have seen. Were we basically fertilizing away any potential negative effects of elevated CO2 on plant pollen or pollen protein. So I worked with an undergraduate named Grace, who's pictured here, to conduct another follow-up study where we looked at the role of elevated CO2 and nitrogen fertilizer on one plant species. And so we picked borage. We didn't have the results back to know which, if any of the species were going to show, a strong response to this elevated CO2, but we picked borage because it makes a lot of flowers that are pretty easy to collect pollen from. So that was our choice here. And so again, in Grace's study, our ambient CO2 is about 400 parts per million with an elevated CO2 around 700 parts per million. We conducted 3 rounds of plant growth and then collected pollen samples when the plants started flowering. And so we had 4 greenhouse chambers, 2 with elevated CO2 and 2 with ambient CO2. And in each of those, half of the plants experienced nitrogen fertilization and half were not fertilized with nitrogen at all. So hoping to sort of tease apart the effects of nitrogen fertilizer and CO2, at least in this plant species. And again, to our surprise, we found no effect of either treatment on pollen percent nitrogen. So here in this graph, we have treatment across the horizontal axis and percent nitrogen in the pollen on the vertical axis. In the left 2 bars, the orange ones, that's the ambient CO2 treatment, and the right 2 bars that are blue, it's the elevated CO2 treatment, with the darker bars, second and the fourth, being the ones that also received nitrogen fertilization. And so we found no difference across CO2 treatments, nitrogen treatments, or any interaction between carbon dioxide treatment and nitrogen. So here we couldn't affect borage pollen with any of what we were trying to throw at it. And so perhaps we're thinking that because pollen is such an important tissue when it comes to plant reproduction, you need your pollen grains to be viable and available for reproduction, that perhaps it's shielded from sort of these environmental effects. So, leaves are essentially disposable, right? The plant needs that to grow, but it doesn't need it to reproduce. And so if a leaf has lower percent nitrogen, it's maybe going to persist just fine. But if the pollen has reduced percent nitrogen, maybe it's no longer viable and the plant can't reproduce. So that's what we think might be going on here, but not totally sure. A little bit surprised with these findings again. And so if we sort of sum this all up, our conclusions on elevated CO2 across the pollen percent nitrogen and all of the other variables that I measured that I really wasn't able to talk about today, we can conclude really that flowering plant responses to elevated carbon dioxide are really species specific. So there were sort of no general trends across the species we studied. And we found that elevated carbon dioxide does not appear to lead to an overall decline in pollen percent nitrogen, which is a proxy for protein, which is a positive. And we did not find any changes to pollen percent nitrogen levels in borage when we added nitrogen fertilizer. So I think it was a little bit of a bummer to find very few significant findings with this work, but I think it was also kind of hopeful and nice to see that perhaps this aspect of global change is not going to be as devastating for pollinators as I think we thought when we went into this project. So we'll move on from elevated CO2 to some work I've been doing on temperature. And as I mentioned, in the state of Wisconsin, where I am living and watching snowfall from my living room, we see that over time, temperatures in my state are warming. They're getting warmer over time, and they're predicted to continue warming. This is true in many other places around the world, including Canada. And it's a thing that we're all going to have to sort of deal with and figure out how to overcome or mitigate in some way. And we're pretty lucky because most of us have a nice warm home and a winter coat and the ability to sort of change our surroundings so that we're comfortable, right? We can put clothes on, take clothes off, go outside, whatnot. But insects really are vulnerable to these external temperatures. So they don't have the same ability to, thermoregulate with external devices as we do. And previous research shows that temperature does play an important role in memory and retention in insects. So warmer temperatures, like short-term heat waves, can disrupt memory and learning in bumblebees, whereas cooler temperatures can help facilitate memory formation in other insects. So it seems that this, global trend towards warming temperatures and the role that temperature might play in memory could be something important for pollinator foraging and for their success going forward. And so I was specifically interested in understanding whether or not bumblebee learning is affected by the temperature it learns in. And so thinking about kind of, is there sort of an optimal learning temperature for this species of bumblebee? And this is work that was done in collaboration with Taylor Wartell, who was an undergrad at the time, and then Acacia Tang, who was a technician but is now a PhD student at UW-Madison. So in this study, we wanted our bumblebees to learn that their less preferred flower color has higher rewards, which in this case was twice the sugar than flowers of their preferred color. And so we built a little arena with an infrared camera, and we gave the bees the choice between yellow flowers and purple flowers. And then we recorded their foraging. And what Acacia did was create a computer vision tracking program that allows us to basically automatically detect our bees as they forage in this arena. And so you can see there's a little dot following the bee around and a little outline around her, showing where she's going, and the color changes when it indicates that she's foraging. This was not perfect for a couple of reasons. If the bee held too still, we sort of lost it in the tracking, but it could also go underneath these 3D printed flowers and sort of fragment into multiple chunks of bees. And so we were pretty successful in getting the bees to forage and to start to look at this question, but we thought we could do it better. So we designed a forage box 2.0. That's what we called this. This was our forage box. And so we designed this larger arena that allowed the bees to make more choices. So we used smaller flowers and gave them a lot more. And we also used a much smaller volume of nectar. So in the previous study in the video we were just watching, the bees basically had nectar wicks full of sugar water to interact with and could basically fill themselves up with 2 or 3 flower visits. And so then they would just sort of give up on foraging. And so with this arena, we had a built-in camera with this Raspberry Pi attached to it. And then we were able to get the bees to forage for much longer when they had to sort of visit multiple flowers to fill up. The other thing we added for the second round of this was a training arena. So when you put bumblebees in this weird shiny white box full of fake flowers, they don't really know what to do with it unless they've been exposed to that sort of an environment before. And so to encourage them to forage more readily and more often, we hooked up the colonies to this training arena. And so they were allowed to forage on these half blue, half yellow flowers for a week before they were exposed to their sort of testing in the second phase of this study. And so we, with this Forage Box 2.0 study, we're trying to figure out whether or not the temperature that this bumblebee learns in, and here we use the common eastern bumblebee, Bombus impatiens, and so whether the temperature that they learn in really dictates their success foraging in other temperatures. And so the setup for this is that we would give bees, a 10-minute foraging run on day one. That was their sort of learning trial, and then a testing trial on day two. And so we had 3 temperatures in each of day 1 and day 2, 18, 25, and 32C. And then we had 9 total combinations so that we were able to test, all of the day 1 and day 2 combinations. And so from this, we were able to collect data on the amount of time the bees spent foraging on blue versus yellow flowers, the number of flowers they visit, so how many blue flowers, how many yellow flowers are they visiting, and the proportion of each and whether or not that's changing over time. How quickly they found their first yellow flower, and the color of the first flower they visited. So that's sort of indicating, when they're a little bit naive, are they preferring blue or are they preferring yellow? And so here's what that looks like in the Forage Box 2.0 setup. So we also had improved computer vision tracking tools. So you can see the bee is being followed around by a white dot on her back, which will turn yellow when she starts foraging on yellow flowers and blue when she starts foraging on blue flowers. And so this is about 5 minutes of foraging, sort of condensed into a minute or two. And I think she visits a blue flower next, so we'll just give her another second. Yep. So this is how we were able to collect our data, and that basically helped us to streamline the data collection process so that we didn't have to sit and record each individual foraging bout on our own. And so I have some preliminary results. I'm still in the process of getting these analyzed, but I have 2 sets of data that I can show you. So this set is first bees that learned in 18C and were tested in 32C. And so in the panel on the left, I have the number of flowers visited, and on the right is the duration foraging on each color. So how long did they spend in seconds foraging on blue versus yellow flowers? And the blue flowers are denoted by blue bars and yellow flowers by yellow bars. And so if we look at the number of flowers visited on this trial 1, so this is the learning trial, we see that there's about equal visits to both blue and yellow flowers. When we look at the results on foraging duration, we see the same, slightly longer on yellow flowers, but pretty similar. But in day 2, what we see for the number of flowers visited, that these bees are preferring to visit yellow flowers over blue flowers. And so that's sort of what we were hoping that they would figure out. And this is sort of the pattern we hope to see. And when we look at foraging duration, we also see the same trend there, where they're spending less time on blue flowers and more time visiting yellow flowers in day 2. And so here's another set of bees that learned at 25 and then were tested at 18. And so again, pretty even split on the learning trial for the number of flowers visited. But interestingly, we see for this set of bees that they were immediately spending more time on the yellow flowers and the blue flowers. When we look at what happened in the testing trial on day 2, we see the same pattern as the previous set of bees, where we have more visits to yellow flowers than to blue flowers, and that's sort of the number of blue flowers visited dropped off in day 2. But interestingly, the foraging duration didn't change between days 1 and day 2. So from this work, we're hoping to sort of better understand how temperature affects foraging and learning in bumblebees, thinking that, if bees might learn perhaps less efficiently at warmer temperatures, that a warmer future could reduce their foraging efficiency. And that perhaps if the temperature in which the bee learns in dictates their performance in other temperatures, that these kind of more volatile day-to-day temperature swings may not actually disrupt foraging efforts. And so where that sort of comes from is maybe it's similar where you are in Toronto, but in the Midwest, especially in spring and in fall, we can have pretty big temperature swings day to day. You know, it might be 24 Celsius one day and 2 and snowing the next day. And if you're a bumblebee queen that decided to come out on that warm day, you know, having this sort of pretty big swing could influence your foraging. And maybe if you learn on the warm days, it will be perhaps not problematic. So hoping to dive into this a little bit more and to hopefully expand this to look at more bumblebee species and just more pollinator taxa in general to sort of understand a little bit bigger picture how temperature influences foraging across sort of pollinators in general. So I'll move on to the last third of my talk and talk a little bit about some of the work I've done trying to understand how microhabitats within the landscape influence bee foraging. And so the previous study I talked about was done in the lab on a very kind of small controlled scale. And this is a study looking at sort of temperature and microhabitats, but out in the field. So kind of complements the previous work. So you're probably tired of me telling you that, you know, the globe is changing. You know, things are changing in the way that we live. And but I'm going to say it one more time because that's what we're talking about today is global change in pollinators. I was really hoping to find a nice map here of either, you know, the US and Canada or the whole of the North America to talk about, you know, global change, but I could really only find a good map of the US. And so what this map is highlighting is sort of all of the ways that we're expecting the United States to change in going into the future. And so one of the ways, again, that we know that our world is changing is that it's heating up. And so I've been sort of thinking about, you know, in the field, how do pollinators cope with these warming temperatures? And do they have any sort of strategies or tricks to sort of help them better deal with these warming temperatures? And this project, again, was helped by Taylor Wortell and Rafael Salas, pictured here. And Rafael is a technician who has been working in the Crawl Lab where I did my postdoc for a couple of years. And so here I set out to answer the question, is pollinator foraging behavior altered by thermal microhabitats within the landscape? And sort of the thought behind this came from something that's become quite popular here in the US is first installing solar arrays over pretty large swaths of land and then planting them with pollinator plantings or prairie plantings underneath. And so I don't know if this is popular in Canada as well, but you know, you have all this habitat, and because those panels don't move, you have a chunk of potentially available habitat that's going to be shaded for part or for all of the day. And you know, when you sit under a shaded tree, it's a little bit cooler. And so I'm thinking, you know, maybe pollinators in a situation like this where they have, you know, dedicated shade might also rely on that cooler area during the heat of the day. And I think when we do a lot of work looking at pollinator foraging in the field, we sort of preferentially pick days that are warm and sunny. You preferentially pick flowers that are in the sun and not in the shade. And so that it's something that we sort of like select against automatically when we're doing a lot of this type of work. So just trying to think about whether or not shade plays a role in foraging at all. And so I had a couple of predictions in this study. First, that when temperatures are hot, visitors would prefer shaded flowers. And that when temperatures are cooler, visitors would prefer the sun flowers. And this is like sunny flowers, not sunflowers, helianthus. If we look at that sort of over the course of a day, I think it might look something like this, where sort of as the sun is coming up and the sun is going down, we're going to have visitors that are preferring these flowers that are in the sun. But during the heat of the day, the visitors would actually prefer the flowers that are shaded because it provides them a little bit of a thermal refuge. So it's a little bit cooler and the flowers are, you know, a reprieve from the heat of the day. And so to test this, I used a field plot where I had 16 2x2 meter plots, each with 2 meters in between. And I had pots of this dwarf zinnia that I put in each of these 16 plots. For 8 of these plots, they were randomly selected to be shaded using a 30% shade cloth. That was basically as shady as we could go without influencing plant growth in any way. And so we use these BioQuip cages that are about 2 meter by 2 meter steel frames that are typically used for sort of field crops with this sort of tan mesh over the top. And so we just use the frame, not the tan mesh, and put our own shade cloth over the top and seeded each of the plots with potted zinnias like we see here. And so this is what the field setup ended up looking like. So we had our 16 plots here, and you can see the shade treatment on the ground. So each of the cages with the shade cloth, and we tried to position the shade cloth so that it would keep the shade, under that specific 2 meter by 2 meter block of land as much as possible. To record our flower visitors, we used automated pollinator camera traps. So you can see in this image on the right, there's a little camera pointing at a flower that's being visited by a drone fly. And each of these camera traps, we installed 8 of them in this study. So you can see each solar panel in the figures on the left, that each one of those is a camera trap, each equipped with 4 cameras. And so to measure pollinator visitors, again, we use these camera traps, just like you might do if you wanted to know if you had, say, coyotes in your backyard, or if you were going to go deer hunting, like where you might look for deer. You'd set up a wildlife camera trap that would just passively collect images. And so we use effectively the same thing, but scaled down for insects. And so we've been calling this the AutoPolls, which stands for Autonomous Pollinator Sampler. And it is run using a Raspberry Pi, which is I mentioned earlier, is a single board computer, and you can program it to do a handful of things. Here, it's also connected to 4 borescope cameras, and a borescope camera is essentially what a plumber would use to look for, a clog in your drain. So it's on about 15 feet of kind of a stiff wire, and it's already waterproofed, meaning that it's ready to go out in the field, and we don't have to worry about any issues with like weather or things like that. Along with each borescope camera, we have a thermocouple wire. So this blue wire that you can see wrapped around with it. And then there's a little black circle on the end. That's what we call our operative temperature model. And so that's a bit of Scoopy clay that's about the same size as a bumblebee to sort of mimic what a bumblebee would feel if it was outside, at that time. And so this camera trap packs up into a junction box that's waterproof. We can set it outside. It's connected to a battery, which is connected to a solar panel, so it can sit outside for weeks at a time. And then we use this to record pollinators from 5 a.m. to 9 p.m. for 15 minutes of each hour. So 5 minutes on, 15 minutes off throughout the whole day. And each camera can record approximately 1 image per second. And then we also paired this with some human observations in 5-minute intervals. So once we put the camera traps out, we're going to end up with a boatload of images. And so to figure out how to get all of those images into something that's a usable data set, we walked it through a couple of kind of machine learning pipelines. So we collected our images, then we ran our images through the universal insect detector. And what that does is it separates all of our insects basically, or all of our images, into 2 piles, right? Where we have images with insects in one and images without insects in another. And so we basically just discard all of the images that don't have insects and abbreviate our data set to those that do have insects. Once it's going through and scoring whether or not it thinks there's an insect, it's going to give you some sort of a degree of confidence. So it might think that there's an insect like we see here, and it might be very confident that there's an insect, but it might also see some other weird plant part or something that's not an insect and think, well, there's a 20% chance that this could be an insect. So we have sort of a range of confidence in the universal insect detector's, confidence in its ability to detect insects. So we get rid of all of the images that don't have insects in them. And now we take that subset of images with insects, and we need to figure out which insect is which. So to classify these insects, we use Bee Machine, which was developed by Brian Spiesman at Kansas State. And what this will do is it will give us an identification. So here it might think that this is a halictid, and it might be 80%, 87% confident that it's actually a halictid. So we get this classification of the insects. So first we figure out if there's an insect, then if there's an insect, who it is. And then you have essentially a workable data set. So in this case, it looked something like this. We collected data for 6 weeks in 2023, so July 14th through August 31st. And all of these images on the right are images from the camera traps themselves of things that we detected visiting our flowers. So for a plumber's camera, I think it does a pretty good job of capturing, identifiable high quality insects, which is great. So in total, over 6 weeks, we collected a little over 5 million images. And again, this is where we use that computer vision pipeline because I sure don't have the time to go through that many images in great detail. We ran this data set through the insect detector, and we found that about 900,000 images actually contained insects, and that in total, a little over 1 million insects were detected in this data set. So there were situations here where there were multiple images or multiple insects per image. If we filter for just the high confidence detection, so the situations where we feel pretty confident that we're actually seeing an insect, we end up with about 13,000 high confidence detections. So it takes the data set down quite a lot. When we look at the high confidence detections with the high confidence identification, so first we're sure we have an insect, and then we're pretty sure that the identification is right, we end up with about 2,200 detections. And then sometimes insects might spend more than 1 second visiting a flower, and so may be detected, sequentially multiple times. And so to avoid sort of overcounting those situations, I clustered visits by time. So for example, if a honeybee visited a flower for 10 seconds, that would be counted as 2 visits instead of 10. So just helping to sort of reduce the overcounting of our visits. So the total data set for this ended up being about 1,100 visits. And those 1,100 visits were primarily made by beetles in the order Coleoptera, so that's here in green. And then the next most common visitors were true flies, Diptera, and then syrphids, so hoverflies, a family of true fly. And then we saw a whole bunch of different bees, a few lepidopterans, most commonly this Melisodes bimaculatus and Bombus vagans, which makes sense with my own observations out there. And when we compare that to what the humans saw, it's pretty similar. So the top 3, the beetles, the hoverflies, and the other flies, they matched the top 3, not in exactly the same order, but still about the same. And the other bees and whatnot that we were seeing matched pretty well. The only thing that's really different between these two is there's this sort of other bar in the human observations, and those were things like true bugs in Hemiptera that are not included in the insect identification model that we used for the image data. And so now if we look at our detections across the time of day and across temperature to sort of understand whether or not we are seeing that sort of temporal shift in preference, we can first look at temperature across time of day. So first confirming whether or not our shade treatment did actually reduce the temperature in the heat of the day. And if we plot time of day across the horizontal axis and temperature in Celsius across the vertical axis, we do see that from maybe about 11:30 up to about 3:30 or so, that the temperature in the sun treatment is higher than what we saw in the shade treatment. So that's good. Our shade treatment worked, and we were able to reduce the temperature of those flowers in that camera. And so then we can look at what visitation over the time of day looks like. And so remember, this is what I predicted, right? We had sort of more visitors first thing in the morning to the sunflowers, and again sort of at the end of the day, but then sort of in the heat of the day we would expect there to be more visitors on the shade flowers. And so when we look at visitation, it looks like this. And so it's not exactly kind of what I predicted, but it's pretty close. So what we see is sort of in the first half of the day, when temperatures aren't too different between treatments, that there's basically complete overlap between the number of visitors to sun versus shade flowers. But then sort of towards the heat of the day, we do see that the flower visitors are utilizing the shade flowers more heavily than the sunflowers. So that was pretty cool. I was not really expecting to actually find this, just given all of the existing literature on only using sunflowers for pollinator research. So I thought this was pretty cool, and I think that really the takeaways from this work is that first of all, our shade treatment was successful, which is great. And then when our temperatures really were hottest during the day, the shaded flowers were more heavily utilized by flower visitors than the sunny flowers. And so I think to me what this says is that when we're considering developing habitat enhancements and pollination or pollinator conservation efforts in the future, that it could be really wise and helpful to consider potentially the role of thermal refugia when designing these. So with that, I thank you all very much for listening. And if we have time, I'd be more than happy to take questions.
Thank you so much. Excellent talk. A lot of it's unpublished too, which is lovely. So we look forward to seeing that when it comes out.
Yeah, I think it's all unpublished. I thought about talking a lot about my work from Australia, but I thought that maybe
No,
some
this
new
is
things would
exciting.
be more
Yeah.
interesting. Yeah.
Yeah. And what we can link as well as the stuff does become available, we can put those links alongside the YouTube to update that. But so excellent talk. I have many questions, but I'll open it up to the audience because we already have many in the chat. Lawrence Packer says, thanks for an enjoyable talk. I wonder about the effect of elevated carbon dioxide on plant fitness, uh, which will also impact pollinators, albeit as immediately as pollen quality might.
Yeah. I think that's a really good point, and that's something that I've been thinking about too. Yeah, so thinking about whether or not elevated carbon dioxide is going to influence, just basic plant reproduction and plant fitness, and how that might influence pollinators ultimately. I think if I was going to do that study again, we sort of, a little bit of background on that study is we did it during the winter time in Wisconsin, and we didn't have a great place to try to grow plants with elevated CO2 because the greenhouses were not air conditioned. So when it was too warm, we had to just open the windows. And so we sort of had a very small window when we could do this study and make sure we could keep the CO2 levels where we wanted without like just letting it all go. And so I think if I was going to do this study over again, I'd try to find a better environment to grow them in so we could do the study for longer. Because I think one of the things that I'm still really interested in and wondering about is like seed set and plant fitness in that regard. Are they still able to reproduce, and are flowers producing the same number of seeds across treatments or not? So yeah, I think that that's a really good point, and something that, yeah, I've still been considering as I've been digesting all of this work on carbon dioxide.
Great, thanks. We have another question. Amelia Morin asks, what software was used to track the bumblebees?
Yeah, so that's a good question. It was actually custom written software by Acacia. So Acacia was using background subtraction, so basically looking at change from frame to frame to basically figure out, whatever was changing from image to image would be the bee. So that's custom software that we used and made to track the bees. And if you are interested, you can send me an email and I could potentially connect you with Acacia. I know they've just started a PhD position, so don't have a ton of availability, but could definitely point you to some resources or share that code with you if it might be something useful for you.
Is that a software that might become published, or is that, anything about that, or?
That's a good question. I don't know. I think some of it will be, well, yeah, probably the code would be published um,
Their work, yeah.
with that paper, but I don't know that it'll be published sort of as its own separate tracker program, if that makes sense.
It could be. Is it like an R-based thing, or what kind of software? Do you know? Background?
It's Python, and so yeah, so we'd share the code with the publication, but I don't, yeah, you could use the code yourself and with the Python packages that are included with that.
Yeah, no, it would be hugely useful. I think some people underrate their own code, and that would be it could be a publication unto itself, is my point. And
Yeah.
but yeah, if there is any movement there, but yeah, if anyone wants to connect, there's the email here on the screen. So please do reach out for software, and because this is so much unpublished work, it's nice to see. Because sometimes, yeah, we get a bunch of published stuff, which is great, but we could have already read those papers. So this is exciting because this is really at the forefront of a lot of cool tech here.
Yeah, and I think I'll like also if you have questions on tech in general, like I feel like I came to a lot of this as the entomologist using the tech and have been able to sort of develop the skills to use some of these fun new electronics, but that I definitely didn't have this background before starting my postdoc. So I
Yeah.
think one of my strengths is being able to explain this to other ecologists and entomologists that maybe don't have computer science training. So yeah, totally happy to answer questions or to, talk through things with anyone if you're interested.
Great. Yeah. It's always it's a barrier to entry for some. So yeah, you're a nice kind of middle ground. I don't see any more questions in the chat, but I had one about the nutrition, your first stuff. I think that was actually published already, but the idea of looking at total nitrogen is a great metric, but I wondered about, because we've been doing some pollen nutrition analyses as well in our lab. Have you at all looked at other variables like, there's a body of literature on free versus bound amino acids, essential amino acids, pollen lipid ratios. So nutrition can kind of be on a gamut. As you can look at CO2 and its complexity. Protein itself is a kind of a complex biomolecule. So I was wondering if there was any additional data or future direction that might unpack that further, or were you just kind of going to leave it at the total nitrogen story for now?
There's a little bit of data in that publication. We did look at pollen metabolomics for, I think, 3 species. And we did find that, I'm not going to be able to remember exactly off the top of my head. There were some things that in the metabolomics that did change. I don't think that, the amino acids we looked at were one of the ones that changed. But yeah, that's another thing that I think if I could do the study over again, I would want to do a lot more complex nutrition analyses than just percent nitrogen. That's what we sort of set out to do initially. But I think that the more I've dove into this literature, yeah, like protein to lipid ratio is really important and thinking about like the specific compounds. So if maybe total nitrogen is not changing, but some important components like amino acids are, then that would be good to know as well.
Is there any plan to do that or not at this time?
Not with that data set. The thing that I think that I'd like to do going forward would be to try to get pollen from face experiments. So the free air carbon exchange experiments where they're able to basically like pump the carbon dioxide around existing plants, because keeping the CO2 elevated was really our biggest challenge in a greenhouse.
Mhm.
And so working with plants, and so there's a research station not far from here, and I think that that would be a really cool project for some undergrads is to collect some pollen from some of those flowering plants and look at whether or not other aspects of the pollen chemistry has changed. So I don't have immediate plans, but it's something that's stewing in the back of my mind for sure.
Great. Alrighty. Well, yeah, we're on the hour. So I think with that, we will wrap this up. Again, here is the email address and social. And again, we'll be on YouTube shortly. And kick it back to Laura for a second.
Wonderful. Thank you again, Olivia, for your wonderful talk. Always, as Sandra said, always great to see such preliminary data being presented because it's very exciting to see. I just wanted to remind everyone that we do have another speaker series talk planned for next month, Friday, February 21st, again at 11 a.m. This will be with Dr. Nadia Svetkov. You can register for this on our website. And with that, I would like to thank you all very much for joining us today for the BC Speaker Series, and we will see you hopefully again next month.